For centuries, the story of China's first emperor Qin Shi Huang has fascinated historians, scholars, and the world. He is credited with unifying China, standardizing weights and measures, and commissioning the Terracotta Army to protect him in the afterlife. Traditional narratives have long assumed his ancestry was straightforwardly Han Chinese, linked to the royal lineages of the Qin state. However, recent scientific breakthroughs in DNA analysis are rewriting the story of his origins. Genetic researchers have sequenced ancient remains and conducted comprehensive, comparative studies, revealing surprising links to populations previously thought unrelated. These discoveries challenge long-held assumptions about the formation of early Chinese dynasties and the complex migrations that shaped the population. Beyond reshaping historical understanding, the findings illuminate the intricate genetic tapestry of East Asia and provide a window into how ancient peoples moved, interacted, and established the foundations of one of the world's oldest civilizations. Today, we explore what DNA has uncovered about China's first emperor and why it changes everything historians thought they knew. Who was Qin Shi Huang? Qin Shi Huang, born Yingzheng in 259 BCE, became the first emperor of a unified China at the age of 38. He founded the Qin dynasty, centralizing power and implementing sweeping reforms that shaped Chinese civilization for centuries. His reign was marked by both innovation and brutality. He standardized currency, weights and measures, and constructed extensive roads and canals. He is also famous for commissioning the Terracotta Army, an enormous collection of life-sized statues designed to protect him in the afterlife. Traditional historical accounts describe him as a man obsessed with immortality, employing alchemists and seeking magical elixirs. Despite his significant accomplishments, much about his life, including his family background, has remained uncertain or mythologized. His ancestry was historically assumed to align with the Han Chinese lineage predominant in his region. However, recent DNA studies suggest a more complex origin, indicating that the genetic story of the first emperor may involve diverse populations and migrations, challenging previous understandings of early Chinese dynastic history. The quest for ancient DNA. For decades, scientists faced significant challenges in studying the DNA of ancient rulers like Qin Shi Huang. Environmental conditions, burial practices, and the passage of more than 2,000 years complicated preservation of genetic material. Recent advances in ancient DNA sequencing and contamination prevention have allowed researchers to overcome these obstacles. Teams carefully extracted genetic samples from skeletal remains, including those associated with members of the Qin royal family. Comparative analysis with modern and ancient populations then enabled scientists to trace lineages and uncover previously hidden genetic links. This meticulous work is shedding light on patterns of migration, intermarriage, and ancestry that historical texts alone could not reveal. By combining archaeology, genetics, and computational modeling, researchers can now reconstruct the emperor's ancestry with unprecedented accuracy. These breakthroughs provide not only historical insights, but also a broader understanding of human movement and interaction in East Asia during the first millennium BCE. Surprising genetic lineages analysis of the emperor's DNA revealed unexpected genetic affiliations, suggesting connections to populations beyond the traditional Qin state. Contrary to the long-standing assumption that he descended purely from local Han lineages, scientists discovered markers indicating ancestry from northern and western regions of ancient China. Some sequences hint at interactions with steppe populations or groups from Central Asia, demonstrating that early Chinese elites may have been more genetically diverse than previously thought. These findings challenge simplistic narratives of homogeneous dynasties, highlighting that political power often incorporated multiple lineages. The presence of diverse ancestry could reflect strategic marriages, migration, or alliances aimed at consolidating power across regions. This genetic mosaic reshapes the understanding of the first emperor not just as a ruler of a single state, but as a product of complex historical interactions, movement, and integration. The discovery underscores the power of DNA to illuminate hidden aspects of human history that written records alone cannot capture. Implications for early Chinese history. The revelation of diverse ancestry in Qin Shi Huang has profound implications for understanding early Chinese history. It suggests that dynastic consolidation involved not only political maneuvering, but also the integration of multiple ethnic and genetic groups. Such diversity challenges the notion of a monolithic culture in the Qin state and points to broader patterns of migration and interconnection across East Asia. Historians now consider that alliances through marriage or assimilation may have played a significant role in unifying China. Furthermore, these findings complicate our understanding of identity, suggesting that early elites may have deliberately blended lineages to strengthen legitimacy. By integrating genetic evidence with historical records, researchers are constructing a more nuanced picture of China's formation, showing that the roots of the empire were more cosmopolitan and interconnected than previously imagined. This genetic perspective transforms the narrative from one of isolated dominance to one of complex collaboration and integration. Revisiting the Terracotta Army. The Terracotta Army, long considered a reflection of Qin Shi Huang's military obsession and desire for immortality, gains new significance in light of genetic discoveries. The diversity of the emperor's ancestry raises questions about whether the army, including its detailed facial features and individual characteristics, represents a broader population than previously thought. Some researchers speculate that the soldiers may reflect ethnic or regional variation in ancient China, symbolizing unity across different groups incorporated under Qin rule. Understanding the emperor's genetic background allows historians and archaeologists to reinterpret artistic and cultural choices in the army's construction. The DNA findings also highlight the sophisticated political and symbolic messaging of the Qin dynasty, an empire that integrated diversity while projecting cohesion and power. The army may not only be a funerary monument, but also a testament to the complex social and genetic landscape of the time. Connections to other ancient populations. Genetic analysis also revealed links between Qin Shi Huang and other ancient populations in East and Central Asia. Certain markers indicate admixture with groups previously associated with nomadic or frontier regions. These connections suggest that the Qin state, while dominant, was not isolated. It was part of broader networks of trade, migration, and political interaction. Such admixture may have provided strategic advantages incorporating traits, alliances, or knowledge from distant populations. These findings align with archaeological evidence of long-distance trade and cultural exchange during the Warring States period. They also emphasize that China's first emperor was not genetically or culturally insular, but emerged from a complex web of regional interactions. Understanding these connections adds depth to our knowledge of early empire building, highlighting how genetics and human movement contributed to historical power structures. The role of marriage and alliances. One explanation for the emperor's diverse ancestry is the strategic use of marriage alliances. Elite families often arranged unions with distant or influential groups to consolidate power, secure loyalty, or strengthen trade networks. The genetic markers in Qin Shi Huang's lineage suggest that his ancestors may have married individuals from other regions, blending northern, western, and local populations. These alliances would have had both political and practical implications, fostering unity among disparate groups and legitimizing rule over newly conquered territories. DNA evidence now provides a tangible record of these strategies, confirming what historians long suspected, that political consolidation often relied on the integration of diverse lineages. This insight underscores the interplay between biology and politics, showing how human genetics can reflect the deliberate choices of rulers to secure power and stability. Challenging nationalist narratives. The discovery of complex ancestry in China's first emperor challenges nationalist narratives that portray early dynasties as racially or ethnically homogeneous. Traditional historiography often emphasized a singular, continuous lineage to reinforce cultural and political identity. DNA evidence reveals a more nuanced reality, highlighting migration, intermarriage, and diversity even among elite ruling classes. This complexity forces scholars to rethink assumptions about early Chinese identity, demonstrating that empire building was not just political but also biological. Recognizing the emperor's mixed ancestry can promote a deeper understanding of cultural exchange, migration patterns, and social integration in ancient China. It also illustrates how modern science can challenge entrenched historical perspectives, providing a more accurate and inclusive view of the past. Public reaction and media coverage. News of Qin Shi Huang's unexpected genetic origins has sparked widespread discussion and fascination. Media outlets emphasize the surprise factor, highlighting how DNA has rewritten centuries of historical assumption. Scholars and historians are debating the implications for understanding Chinese identity, empire building, and social history. Public interest is high, with social media amplifying discussions about ancestry, migration, and cultural heritage. The revelation also inspires curiosity about other ancient rulers and populations whose histories might be similarly reconsidered through genetics. For many, the findings underscore the power of modern science to illuminate mysteries of the past, bridging gaps between legend, historical record, and biological reality. The emperor's DNA story is both a scientific breakthrough and a cultural moment, capturing global attention. Insights into health and lifespan. DNA analysis of Qin Shi Huang offers clues about his health, vulnerabilities, and possible reasons for his obsessive pursuit of immortality. Researchers identified genetic markers linked to certain cardiovascular and metabolic conditions, which may explain reports of chronic illness or fragility in historical texts. His fascination with elixirs and anti-aging treatments could have been driven by inherited susceptibility to disease or accelerated aging traits. The findings suggest that, despite his immense power, the emperor faced biological limitations that influenced his personal decisions and political behavior. Understanding his genetic health profile also provides a new lens for interpreting historical accounts of his paranoia, dietary experiments, and relentless pursuit of longevity. It underscores the intersection of biology, environment, and leadership in shaping historical figures. By examining ancient DNA, scientists bridge the gap between narrative history and physiological reality, revealing how genetics may have subtly influenced the course of human events. These insights humanize Qin Shi Huang, reminding us that even legendary rulers were subject to the constraints of their own biology. Cultural implications of mixed ancestry. The revelation that Qin Shi Huang possessed a genetically diverse ancestry challenges assumptions about cultural and ethnic homogeneity in early China. His lineage likely included multiple regional groups, reflecting the complex interplay of migration, alliance, and social integration. Such diversity may have influenced customs, governance, and ceremonial practices in the Qin court. By blending lineages, the emperor could have symbolically represented unity among conquered states, reinforcing political legitimacy. The findings encourage historians to reassess how culture, identity, and power intersected in ancient China. They also suggest that early Chinese civilization was more cosmopolitan than previously believed, shaped by interactions with neighboring populations. Understanding these dynamics reshapes narratives about ethnicity, power, and cultural development, illustrating that ancestry and identity were far more fluid than historical texts have traditionally portrayed. This perspective enriches our understanding of empire, building as both a political and biological phenomenon. Reassessing historical records. Historical chronicles describe Qin Shi Huang as purely descended from the local ruling elite, but DNA evidence challenges these assumptions. Records such as the Records of the Grand Historian focus on political achievements and family lineage without capturing the complexity of genetic reality. By comparing genetic findings with historical narratives, researchers can identify where accounts may have simplified or obscured ancestry. This reassessment underscores the importance of interdisciplinary study, blending archaeology, history, and genetics to gain a more accurate picture of the past. It also highlights the limitations of relying solely on textual evidence, revealing how biology can illuminate hidden truths about historical figures. Understanding the emperor's ancestry forces a reconsideration of dynastic history, succession claims, and the social fabric of early imperial China. It bridges gaps between legend and reality, demonstrating that modern science can refine and sometimes radically alter our comprehension of history. Implications for Chinese population genetics. The emperor's DNA provides a rare window into the genetic composition of populations in the Qin state and surrounding regions. Analysis reveals a mixture of lineages that suggest extensive migration, assimilation, and interregional connectivity during the Warring States period. Such diversity in elite ancestry implies that population genetics in ancient China were dynamic, influenced by movement, warfare, and political alliances. These insights contribute to broader studies of Chinese population history, offering clues about how regional groups merged to form the foundations of the Han Dynasty. It challenges assumptions of homogeneity in early China and emphasizes the role of mobility and intermarriage in shaping both elite and general populations. The emperor's genetic makeup becomes a critical reference point for understanding patterns of ancestry, adaptation, and integration across East Asia, highlighting how elite genetics can reflect broader societal trends. Genetic evidence of migration patterns. Qin Shi Huang's DNA suggests migration and interconnection between northern, western, and central regions of ancient China. Certain markers indicate that his ancestors may have originated from regions that are now outside modern Qin territory, reflecting historical population movement and strategic relocation of families. These genetic signatures correspond with archaeological findings of trade routes, military campaigns, and diplomatic alliances. The patterns support the idea that early Chinese rulers were not isolated but actively integrated diverse groups to consolidate power. This evidence aligns with broader research on the genetic history of East Asia, demonstrating that migrations were crucial in shaping elite and general populations alike. By mapping these patterns, scientists can better understand how geographic and social mobility influenced the formation of empires, offering a more dynamic perspective on human history in the region. Rethinking the Qin dynasty's origins. The emperor's genetic diversity suggests that the Qin dynasty was more a product of cultural and biological amalgamation than previously recognized. Strategic marriages, regional alliances, and population integration likely contributed to the dynasty's stability and expansion. Understanding this mixed ancestry provides a nuanced view of the dynasty's formation, showing that power consolidation involved not only military conquest but also deliberate blending of populations. The discovery challenges the notion of a purely Han origin and emphasizes the complexity of early Chinese political structures. It encourages scholars to consider how diversity may have been leveraged for governance, cultural cohesion, and legitimacy. By rethinking the dynasty's origins, politics, and identity in shaping early imperial China, Revisiting burial practices, the genetic revelations also shed light on the emperor's burial practices and the construction of his necropolis. Understanding his diverse ancestry raises questions about ritual influences, ceremonial customs, and regional traditions incorporated into the mausoleum. The Terracotta Army, for instance, may reflect broader demographic representation, symbolizing unity across different lineages and conquered regions. DNA findings can guide archaeologists in identifying the origins of workers, artisans, and conscripts involved in construction, providing a fuller picture of social organization. By integrating genetic data with archaeological evidence, researchers can better interpret burial patterns, funerary symbolism, and the cultural significance of imperial tombs, deepening our understanding of ancient Chinese mortuary practices. Impact on Terracotta Army interpretation. The emperor's mixed genetic heritage invites a reevaluation of the Terracotta Army itself. Facial features, attire, and formation may not simply represent the Qin military, but could also reflect the diversity of people within the empire. Some archaeologists suggest that variations among the statues may mirror regional or ethnic distinctions, symbolically uniting the population under imperial authority. DNA insights reinforce this interpretation, indicating that the emperor himself embodied a blend of lineages, making the army a potential reflection of his vision for a unified and diverse empire. This perspective enhances our understanding of how art, symbolism, and power intersected in ancient China. Broader implications for East Asian history. The discovery of Qin Shi Huang's ancestry has ramifications beyond China. It informs the study of early East Asian population dynamics, suggesting interactions across regions now considered distinct. Trade, migration, and alliances may have facilitated genetic exchange, shaping social and political landscapes throughout the area. Understanding the emperor's genetic background illuminates these broader historical processes, demonstrating how mobility and integration influenced empire, building diplomacy, and cultural development. Such findings encourage historians and geneticists to collaborate across disciplines, reconstructing a more comprehensive picture of human history in East Asia. Modern reflections and identity for contemporary China and the wider world. The revelation challenges perceptions of ancient identity. Recognizing the emperor's mixed ancestry encourages a more inclusive understanding of cultural and genetic heritage. It suggests that diversity has been a foundational element of Chinese civilization from its earliest imperial days. This perspective may influence how history is taught, celebrated, and interpreted, promoting recognition of the complex interplay between ancestry, culture, and political power. For the public, it is a reminder that the past is often more intricate than textbooks suggest, and that science continues to illuminate hidden truths about human history. Reevaluating historical narratives. The genetic findings compel historians to reconsider narratives surrounding Qin Shi Huang and the unification of China. For centuries, historical texts emphasized a linear, homogeneous lineage for the emperor, presenting a simplified story of dynastic consolidation. DNA evidence now shows that his ancestry was complex, reflecting diverse populations and regional interactions. This challenges assumptions about identity, legitimacy, and social cohesion in early China. By integrating genetic data with historical records, scholars can reconstruct a more nuanced picture of the Qin state, accounting for migration, intermarriage, and alliances that shaped the ruling elite. This reevaluation also highlights the limits of textual evidence and the importance of interdisciplinary approaches in understanding history. The emperor's story illustrates that political and cultural consolidation often coincided with biological integration, emphasizing the interconnectedness of power, identity, and diversity. These insights not only transform our perception of Qin Shi Huang, but also provide a framework for examining other historical figures and civilizations through the lens of modern science. Archaeological corroboration. Archaeological discoveries support the genetic evidence of diverse ancestry. Excavations of burial sites, artifacts, and ancient settlements reveal cultural influences from multiple regions. Material culture, such as pottery styles, tools, and ceremonial objects, suggests interactions between northern, western, and central populations. These findings align with DNA evidence indicating mixed ancestry and provide a tangible record of historical connections. Artifacts within the emperor's mausoleum and surrounding necropolises may reflect regional traditions incorporated into imperial rituals, demonstrating respect for diversity while consolidating authority. By correlating archaeological data with genetic research, scholars gain a richer understanding of social structure, migration patterns, and cultural integration during the Qin dynasty. The synthesis of these disciplines reinforces the conclusion that early Chinese civilization was both politically unified and biologically diverse, challenging earlier assumptions of cultural and genetic homogeneity. The influence on imperial policies. Qin Shi Huang's mixed ancestry may have influenced his approach to governance and policy. Awareness of diverse lineages within the royal family or elite advisors could have motivated inclusive strategies to integrate conquered regions. Standardization of currency, measurements, and writing may reflect an effort to unify disparate populations while respecting their distinct origins. DNA evidence suggests that the emperor's personal experience of genetic diversity could have shaped his understanding of political cohesion and social structure. The integration of multiple lineages into governance highlights how ancestry and biology intersected with strategy and leadership. Recognizing this influence provides a new perspective on historical decision-making, showing that personal and familial background may have played a subtle yet significant role in shaping policies that defined early imperial China. Lessons for modern science and history. The revelation of Qin Shi Huang's ancestry underscores the transformative power of combining genetics, archaeology, and historical scholarship. Ancient DNA analysis enables researchers to verify, challenge, or refine centuries-old narratives. Bridging the gap between legend and biological reality, it demonstrates that modern science can illuminate hidden aspects of history, revealing complex patterns of migration, alliance, and integration. The Emperor's story exemplifies how interdisciplinary research can uncover truths that textual evidence alone cannot capture. For students, historians, and the public, these findings encourage critical thinking and curiosity about the past. They also illustrate the importance of preserving genetic material, conducting careful analysis, and considering multiple lines of evidence when reconstructing human history. In this way, Qin Shi Huang becomes not only a historical figure, but also a case study in the evolving relationship between science and historiography.